topic on liposomes, preparation, evaluation, and application. This is part 2. In the previous class, I discussed about the liposomes. What are liposomes, classification, and just started with the preparation. So this is a continuation of the previous part. These are the contents. Here we are going to discuss about the method of preparation, its mechanism, applications, valuation, specifically therapeutic applications, and conclusion. These liposomes can be, the method of preparation of liposomes can be classified basically into two: passive loading techniques, active loading techniques. And these passive loading techniques, again, it can be classified as mechanical dispersion method, solvent dispersion method, detergent removal methods. Again, these mechanical dispersion methods, like under the category of mechanical dispersion methods, the methods like lipid film hydration by handshaking or non-handshaking or freeze drying, microemulsification, sonication, French pressure cell, membrane extrusion, dried reconstituted vesicles, and freeze-thawed liposomes. Second method called solvent dispersion methods. Under that, ethanol injection or ether injection, double emulsion, reverse phase evaporation vesicles, and stable plurilamellar vesicles. Third of the passive loading techniques are detergent removal methods. Here, the detergent removals can be done by these techniques like dialysis method, column chromatography, dilution, reconstituted centivirus and virus enveloped. Now let me discuss each method of preparation in detail. First method, as we mentioned, it is of mechanical dispersion method. Under that, first is the handshaking method, also known as the non-handshaking method. This is the simplest and most widely used method for the physical dispersion. Here, the lipid mixtures, that means it consists of the phospholipids. Different phospholipids I have discussed in the previous class. Various concentrations also have discussed. So the particular phospholipids are selected along with the charge surfacing components dissolved in the solvent mixture of chloroform at the ratio of 2 and methanol ratio of 1, volume by volume is prepared. Then it is transferred into the 250 ml round bottom flask. This flask is attached to the rotary evaporator and it is rotated at 60 rpm. And meantime, this organic solvent gets evaporated at about 30 degrees centigrade or above the transition temperature of the lipids which have been used. So this rotation has been carried at least for 15 minutes until the drug residue appears. Then further, this is evaporated from the vacuum source by closing the lab, and further introducing the nitrogen into that evaporate, and the pressure at the cylinder head is gradually rise during this time, and until there is no pressure difference between inside and outside the flask, the pressure is maintained, and the flask is removed from the evaporator and fixed out to the lyophilizer to remove the further residual solvents. Now you could see this, as already we mentioned, the particular lipids, the phospholipids and the charge surfacing active agents dissolved in the solvent mixture, like as I mentioned, chloroform, methanol, the ratio of 2:1. It is of 250 ml round bottom flask, and it is kept in the rotary evaporator, maintaining the speed of 60 rpm, or it can be done by the hand shaking. So during this process, a film is formed. The film is formed. Here you could see that the film formed. On vacuum, it is dried. Whatever the residue which have been formed, it is dried. The dried film is formed. Again, on addition of or dispersion of the aqueous phase, that means the hydration, again the liposomal dispersion is formed. Again, as it is mentioned, this second stage is that hydration of the lipids. So here, up to here, I discussed up to here the residual solvents. Here the dried films, whatever it is formed, it is dispersed in the aqueous phase. Further, the hydration of the lipids. That means once after releasing the vacuum and removal from the lyophilizer, the flask is flushed with nitrogen. 5 ml of saline phosphate buffer containing solute to be unwrapped is added. Then this flask is attached to the evaporator again flushing with the nitrogen, rotated at the room temperature and pressure at the same speed or below 60 rpm. The flask is left rotating for 30 degrees centigrade until all the lipids have been removed from the wall of the flask and has been given homogeneous milky white suspension free of visible particles. Further, this suspension, whatever the white suspension is formed, it is allowed to stand for 2 hours at room temperature, or it can be a temperature above the transition temperature as a lipid in order to complete swelling process to give the multilamellar vesicles. So this is what it is mentioned in this picture. This is about the handshaking method or non-handshaking method, or it is also called as the freeze drying method. The next method called the microemulsification. Microemulsification, here we use a microfluidizer. This is called as a microfluidizer. Especially this microemulsification is carried, this method is preferred to have a small multilamellar vesicles from the concentrated lipid dispersion. So we want to adopt from the concentrated lipid dispersion small multilamellar vesicles. For such category, prefer microemulsification method. And these lipids, it is introduced in the fluidizers. Either it is in the form of, this is the fluidizer, either it is dispersed in the form of large multilamellar vesicles or as a slurry of unhydrated lipids in organic medium. And these microfluidizer pumps at a high pressure, it is about 10,000 psi, it can maintain, and around 600 to 700 bar through a 5 micrometer orifice. You could see this. The multilamellar vesicles, whatever it is formed, it is passed through this. So to pass through the microfluidizer, which can maintain this high pressure and through the 5 micrometer orifice. Then it is forced into the micro channels, which direct two streams of fluid to collide together at right angles. One is at the high velocity, and thereby it affects the efficient transfer of energy. And whatever the fluid collected, it will be recycled through the pump and interaction chamber until vesicles of the spherical dimension are obtained. After a single pass, the size of the vesicles reduced to a size 0.1 and 0.2 micrometer in diameter. So this is what the microfluidizer, again, it is a continuous process. Next method is called the sonication. Sonication can be done in two ways. There are two types of sonications. The first one is the probe and the other one is the bath sonicator. So let me explain about the probe bath differentiation. The probe, it is mainly for high dispersions. So here we require high energy, but in small volume. That is like a high concentration of lipids or a viscous aqueous phase. Then the probe sonicator is most suitable method. Now, talking about the bath sonicator, it is suitable for large volumes of diluted lipids. Now, probe sonicator, it requires high energy input into the lipid dispersion, but suffer from overheating of the liposome dispersion, causing lipid degradation. So that is a problem with that probe sonicator. Because of the overheating, it leads to the degradation of the lipids, and this sonication tends to release titanium particles into the liposome dispersion, and then it is centrifuged prior to the use. For this reason, because of these disadvantages, bath sonicators are widely used for the preparation of small unilamellar vesicles. This sonication of multilamellar vesicles dispersion is accomplished by placing a test tube containing the dispersion in a bath sonicator, placing the tip of the sonicator in the test tube in a probe sonicator, and sonicating for 5 to 10 minutes above the 3 degrees centigrade of the constant lipid. And this lipid dispersion should begin to clarify to yield a slightly hazy transparent solution. So these particles can be removed by using centrifugation method and thereby we achieve small unilamellar vesicle dispersion. So after spinning, this tube is carefully removed from the rotor with the help of the Pasteur pipette. The liquid with the top clear layer is decanted, leaving the central opalescent layer. That means the opalescent layer will have a small multilamellar vesicles and a pellet behind. And the top layer will be a dispersion of the small unilamellar vesicles. So this you could differentiate. This is the bath sonicator and this is the prop sonicator, where the prop is over here and here is a dispersion which have been formed. And already I told the differentiation of bath and prop sonicator, and with the help of this with the help of this bath sonicator, small unilamellar vesicles will be formed, and how it forms, it's clearly shown in this picture. So initially, the bottom part will be the aggregated lipids. Further layer will be a small multilamellar vesicles, and above layer will be the small unilamellar vesicles. So this is the titanium prop, which I have mentioned over there, the description of the prop sonicator. The next method called French pressure cell method. This French pressure cell method, the purpose of the choice of this French pressure cell method to reduce the size of the particles which have been formed. Initially, the liposomes will be formed. So further, we want to reduce the size of those particles. Then we can adopt this method, French pressure cell method. It reduces particle size by the mechanism of high shear forces. So here, dispersion, as regularly the dispersion of multilamellar vesicles are prepared, and further that is transferred into the apparatus called the French pressure cell method. Here the sample is placed. Here the sample is placed, which is in the form of the multilamellar vesicles, and with the help of the high shear forces, it is converted into small unilamellar vesicles by passing through a small orifice under high pressure. This MLV dispersion is placed in the French press and extruded at about 20,000 psi at 4 degrees centigrade. Once it passes through that heterogeneous population of vesicles, it ranges from several micrometers in diameter to SUV size. Multiple extrusions result on a progressive decrease in the mean particle diameter. Following at about 4 to 5 times passing it, will be able to achieve the particle size of the small unilamellar vesicles. Approximately around 95% of the vesicles have converted into SUVs, and we are able to achieve 30 to 50 nanometer sized particles. Disadvantages: these high shear homogenized need over 10 ml of liposome dispersion to operate properly. This minimum threshold quantity excludes their use in high-cost, small-scale lab operations. Advantage: is more stable as compared to the sonicated liposomes. The next method called membrane extrusion. This membrane extrusion, the size of the liposomes, same like of the previous, it is reduced by gently passing through a membrane filter of defined pore size. So this is what the membrane extrusion method. Here, this is the liposomal dispersion. Here is a polycarbonate filter, where it is able to reduce the pore size. Where it is able to reduce the particle size. And that membrane filter, it is of defined pore size. So this can be achieved at lower pressure than the French pressure cell. And this membrane extrusion technique can be used to process large unilamellar vesicles as well as multilamellar vesicles. In this process, the vesicle contents are exchanged with the dispersion medium during breaking and resealing of phospholipids bilayers as they pass through the polycarbonate membrane. So as I mentioned here, when it is passed through this polycarbonate membrane, the particle size is reduced, and the membrane is of defined pore size. Thereby, it reduces the particle size too. In order to achieve the high entrapment, the water-soluble compounds should be present in the suspending medium during this process. So the material which is not entrapped can be removed subsequently. The next method called dried reconstituted vesicles. It's also known as DRV. Initially the process is that small unilamellar vesicles, whatever it is formed, is dispersed in the aqueous phase. To that, solute also added, further dried by freeze drying, and then rehydration. Further, the film stacks is dispersed in the aqueous phase. Then the solute, whatever it is formed, it may be unilamellar or oligolamellar vesicles. The next method called freeze-thawed liposomes. It is completely based upon the freezing of unilamellar, that is mainly small unilamellar vesicle dispersion, and further thawing at the temperature of at the room temperature at about 15 minutes. So during this time, these freeze-dried lipids are rehydrated with the aqueous buffer. Finally, it is subjecting to the sonication cycle. So during this process, if the small unilamellar vesicles will equilibrate with the solutes inside and outside, and thereby the liposomes fuse, and thereby the size is increased. The size of the liposomes are increased. The second step, sonication, considerably reduces the permeability of the liposome membrane. Perhaps it accelerates the rate at which packing defects are eliminated. So you could see this here. In small unilamellar vesicles, whatever it is prepared, it is dispersed in the aqueous phase. Again, it are to be these solutes. When it is added with the solutes, it is entrapped. And further on freeze drying, either the solutes, it will be in the unilamellar vesicles. You could see the size of it. It can be done by thawing or sonication at about 15 to 30 seconds. Thereby, the freeze-dried product will be obtained. You can see here the lipids are dried over here. Again, by DRV method, either can be rehydrated and or that means it is dispersed in the aqueous phase. Thereby, it may be getting the form of unilamellar or oligolamellar vesicles. And this is the second under the passive loading method. The second method. Till now, I discussed about the mechanical dispersion methods. Here comes the second method called the solvent dispersion method. Under that, ethanol injection and ether injection. This solvent dispersion method, first the lipids, it will be dispersed in the organic solvents. Then it will be in contact with the aqueous phase, and it contains the materials which are to be entrapped with the liposomes. So here again there are two methods. One is with the help of ether, and another one with the help of ethanol. Ether injection. In this method, already we know that solution lipids has been taken, and that has been dissolved in the ether, diethyl ether, which is immiscible with the organic solvents, and is slowly injected into the aqueous phase. Slowly injected into the aqueous phase. Here is the diagram for the ether injection. The lipids are over here. The lipids are dissolved in the diethyl ether, and it is slowly, see here, the lipids are dissolved and is slowly injected. Okay, here the condition is that has been dissolved, it has been injected slowly into the aqueous phase, and further it is encapsulated by a syringe type infusion pump, maintaining the temperature of 55 to 56 degrees centigrade and reduce pressure. So these organic solvents are continuously removed by evaporation at an elevated temperature and reduced pressure. Thereby, it forms a single layer vesicles. Depending on the condition used, the diameter of the resulting vesicles may be 50 to 200 nanometer. So this is the picture of the ether injections. Thereby, it produces the large unilamellar vesicles or single layer vesicles where the size range is of 50 to 200 nanometer. The next method called ethanol injection method. Here also the lipids is dissolved in the ethanol and is injected in the aqueous phase. The difference is that diethyl ether there, here ethanol is injected where into the same. Here the lipids are dispersed in the or dissolved in the ethanol injection, and here it is injected into the aqueous phase. The difference is that this ether injection will be giving slowly, okay, into the aqueous phase, and here it is of a rapid, uh, injecting rapidly. And here, as the ethanol is diluted, uh, the precipitating lipids form liposomes, that is small liposomes, with a diameter size of a small of like of 25 micrometer. It can be formed in the slower injection, resulting in the large vesicles. Disadvantage: it is having low solubility of many lipids in ethanol, limiting the liposome concentration. And also difficulty in quantitatively removing ethanol from the lipoidal bilayer. The next method called double emulsion. So name itself indicates that double emulsification or double emulsion is formed in this by this method. So initially, the organic solution, which have been dispersed with the lipids, and already it contains the water droplet. It is again introduced in the aqueous medium, and further followed by the mechanical dispersion. So during this process, water and oil type of emulsion is formed. Then, on further process, like of double emulsification is formed, that is water and oil in water type of emulsion is formed. So these compartment, two aqueous compartment, has to be separated each other by a phospholipid monolayer. Further removal of the solvent, resulting in the unilamellar vesicles. And we could say that by double emulsification method or double emulsion method, the entrapment can be up to 90%. The next method called reverse phase evaporation vesicles. Here, the aqueous phase and the lipid phase, when it is mixed, water and oil type of emulsion is formed. If there is an excess lipid, you could see here, on further vortexing, multilamellar vesicles are formed. Whereas when the lipid concentration is less, on vortexing is able to produce the large unilamellar vesicles. This is called the reverse phase evaporation vesicles. The next, this is the third method. Till now we are discussing about the solvent dispersion methods. Solvent dispersion methods, under that ethanol ether injection, double emulsion, reverse phase evaporation vesicles, and this is the third method, detergent removal method. Detergents, which are associated with these phospholipid molecules, it forms the hydrophobic portions of the molecule in water. So these structures results in, and it is known as micelles. And here the three stages of interaction for detergents with lipid bilayers can happen. First is when there is a low concentration of the detergents, it equilibrates between the vesicular lipid and the aqueous phase. And stage 2, once after reaching a critical detergent concentration, this membrane structure becomes unstable and thereby it transforms gradually into micelles. And third stage is all lipid exists in mixed micelle form. So these three methods are applied for removal of detergent and transition of these mixed micelles to concentric bilayer form. Here, this detergent removal can be done by the dialysis method. That means the molecules of the detergent are removed from mixed micelle by dialysis. That is by lowering the concentration of the detergent in the bulk aqueous phase. Agents like sodium chloride, octyl glucoside. Next method is column chromatography. This removal of detergent can be achieved by passing this dispersion over a Cephex 25 column pre-saturated with constitutive lipids and pre-equilibrated with hydrating buffer. Example, deoxycholate. So by these techniques, the detergents can be removed completely. Next, I would like to discuss about the mechanism. What type of mechanism is adopted during the preparation of the liposomes? So we know that it is having a hydrophilic as well as the hydrophobic molecules. So these interacts with the lipid, among the lipids, lipid to lipid or lipid water molecules. That is one mechanism. Second, by input of energy like sonication, homogenization, or heating, etc., their energy is developed. Next, by rearranging the bilayer vesicles. For example, initially it might have formed in the form of LUV, large unilamellar vesicles. Then further on adapting the particular method, we are able to reduce the size of these vesicles. Lasik et al. Proposed that symmetric membrane preferred to be flat and energy required to curve them. So sterol determines the membrane curvature. Now, I would like to say that how these liposomes are formed, on what mechanism the liposomes are formed. In aqueous media, phospholipids, you know that the phospholipids are present in this, right? In aqueous media, these phospholipids are not soluble. So they are not soluble and they align themselves in the planar bilayer sheets. Thereby, the lipid cakes are formed, which is thermodynamically stable. So this is the one. We know that this is the phospholipid. The shape is of cylindrical shape. As these phospholipids are not soluble in the aqueous media, it forms a bilayer sheet and it is thermodynamically stable. Whereas the polar head groups face outwards into the aqueous media and the lipid chain of faces towards inwards. The reason is that to avoid the water phase, giving rise to double layer or bilayer. This structure is called as lamella. So thereby, as I told that because of this molecule, these two double layers are formed. And the mechanism I told that when there is an energy, when energy is given by sonication or homogenation or by heating, it starts curving. Curvature of this bilayer sheet occurs. Slightly it is getting curved. Further, the liposomes will be formed. Again, once when it starts curving, upon the hydration, the lipid cakes, already I told what is lipid cakes, this is the lipid cakes. Upon hydration, it swells and they curve completely to form a closed vesicles in the form of spheres. Thus, see here, on hydration, this completely curves and completely rolls, and thereby the liposomes are formed. You could see here the polar head and how the cake, bilayer sheet means this cake formation, how it appears. And again, on hydration, it curves completely and it comes in the form of a liposomes. Again, this is another picture which represents once after the formation of dry lipid film, when it is hydrated, swelling occurs. Again, on agitation, multilamellar vesicles can be formed. Again, when energy is given like sonication or homogenization, either small unilamellar vesicles or by the extrusion technique, large unilamellar vesicles are formed. So this is the in other terms. Here, these are the ratios, the common materials which have been used. These are the ratios which we use for the preparation. Application of liposomes. Liposomes in respiratory drug delivery systems. Already it has proved that the liposomes are highly successful in this category, like of respiratory drug delivery system. Especially it gives sustained release and it prevents the local irritation, reduce toxicity, improve stability in the large aqueous core. Several injectable liposomes are available in the market in this aspect, in the respiratory aspect, for targeting in the lung, ambisomes, fungisomes, myoset, and even the delivery of DNA can also be done through liposomes into the lungs. So these are the areas where the successful rates have been shown in the respiratory drug delivery system, and these are the functions. It is able to obtain all these things. Next, liposome in eye disorders can be used to treat disorder of both anterior and posterior segment, especially dry eyes, keratitis, corneal transplant rejection. In all these categories, it has shown successfulness. Also used as a vector for genetic transfection and monoclonal antibody. And vertipofin is an approved drug for the ocular delivery of liposomes, which is available in the market. Liposomes in the brain targeting. Liposomes, one which is targeted to the brain targeting, it is of small diameter, 100 nanometer, and it undergoes free diffusion and crosses the BBB, blood-brain barrier. And SUVs couples with the brain drug transport vector, thus helping to cross this blood-brain barrier by the mechanism of absorptive-mediated transcytosis. Here, the cationic liposomes more successful. It undergoes this absorptive-mediated transcytosis and endocytosis into the cells. Addition of this sulfatide, a sulfur ester of galactocerebroside to liposome increases the ability to cross the blood-brain barrier. Polysorbate 85 recognized that it enhances significance into the brain. Also, it has been proved that polysorbate 85 can enhance the significance into the brain. Example, amitriptyline. Even it has proved the applications in the anti-infective agents. Liposomes has been used as an anti-infective agent, especially pathogens like protozoal infections, bacterial, even fungal, by targeting the liposomes to the particular residential area into the body. So these are the examples which have been given. These are the active constituents and particular applications for regard of that. Example, annamycin, leishmaniasis disease. And this is by the active targeting approach, and passive targeting approach, same amphotericin B in case of meningitis, leishmaniasis, candidiasis. All these application has been proved in case of the anti-infective agents. Liposomes in tumor therapy. The long-term therapy leads to several toxic side effects. So this liposome therapy, especially in tumor targeting, shows least side effect. Small and stable liposome passively targets the tissue, and it gives a long circulation. Example which have been given here, Doxil. It's a liposomal formulation of doxorubicin. It is prepared by several methods, means Salix and Myoset are the liposomal formulations of doxorubicin. It's used for the treatment of metastatic ovarian cancer. Even it can be used in the breast cancer too. And it's the most available form in the market, Myoset. Liposomes in vaccines. It acts as a liposomes as an immunoadjuvant. It has been acting as an immunoadjuvant. And also it has got some therapeutic applications in the immunological, that is immunological vaccine, and also as a carrier for the immunomodulation, as a tool in immunodiagnostic, not only in the therapeutic, also in the diagnostic side, these liposomes have been identified. It acts by slowly releasing antigen by passively accumulating into the lymph node. Can also be targeted into the lymphoid with the help of phosphatidylserine. Lymphatic targeting with liposomes. Already I mentioned about the purpose liposomes in lymphatic targeting, and is used as a preliminary injection. And here, this active capturing of these liposomes happened with the help of these macrophages in the lymph nodes. And these liposomes, it has uptaken the lymph nodes by the use of biotin. Thereby, it has increased its targeting. It has been used in the lymphatic delivery of methotrexate, and even MRI with gadolinium loaded liposomes is available all in the market. The next is the evaluation of liposomes. So, once the preparations are over, it's very important to know what are the evaluation parameters to be carried out for the liposomes. So, these characterization, basically, it has been identified for physical characterization, chemical characterization, and biological characterization. Physical characterization identify vesicle size, surface morphology, can be done by using TEM or freeze fracture electron microscopy. Mean vesicle size and size distribution, that is submicron and micron range, that is by photon correlation spectroscopy, laser light scattering, gel permeation and gel exclusion, surface charge, it is free flow electrophoresis, electrical surface potential and surface pH, zeta potential measurements, lamellarity by X-ray scattering and 31P NMR, freeze fracture electron microscopy, phase behavior, can be done by DSC, also freeze fracture electron microscopy, and identify the percentage of the free drug and percentage of drug which have been captured. So this can be identified by ion exchange chromatography or mini column centrifugation or radio labeling. And drug release can be carried out by using the diffusion cell or dialysis technique. Chemical characteristics. These are the chemical characteristics which are very important for the liposomes. Phospholipid concentration can be identified by this method, analytical method. Cholesterol concentration by cholesterol oxidase as the NHPLC. Drug concentration as per mentioned in the monograph in the specifications. Phospholipid peroxidation by UV absorbance. Phospholipid hydrolysis, HPLC and TLC, and fatty acid concentration. Cholesterol auto-oxidation, again by HPLC and TLC method. Antioxidant degradation, HPLC and TLC. pH by pH meter. Osmolarity by osmometer. Biological characterization, it's very important, not only the physical, chemical, the biological characterization to be identified once after the preparation, especially sterility to identify the aerobic or anaerobic cultures, pyrogenicity with the help of the limulus amoeba site lysate test, animal toxicity, monitoring survival rates, histology and pathology. These are the biological characterization. Therapeutic application of liposomes. Already it has been proved and is available in the market. These are the drugs and the available forms, its application, particular application and targetability. Conclusion. Liposomes, in fact, it can be used in various range of the pharmaceutical applications. And these liposomes are showing particular promise, especially in case of intracellular delivery systems like ribosomes, proteins, peptides, DNA, etc. And these liposomes have shown enhanced drug delivery, especially to disease locations, by making it to circulate for longer time, and also it is having a better clinical acceptance, and it promotes targeting of particular disease cells within the disease site. Finally, these liposomal drugs exhibit reduced toxicity and retain enhanced efficacy compared with free complements. Only time will tell which of the above application and speculation will prove to be successful. However, based on the pharmaceutical applications and available products, we can say that liposomes have definitely established their position in modern delivery systems.